going to get into the genetics, and this is where things I think get interesting and confusing. And I hope if you get nothing else from this, if you're not going to raise queens, what you get is you start to understand the genetics of the honeybee, because it's totally different than the bean plant genetics that you learned in high school about how Mendel figured this out. So with honeybee genetics, we're talking about haploid genetics, or a haplodiploid system, which I'll explain in detail in a minute. Don't raise your hand and say, I don't know what a haploid is. Remember, in a natural system, without a beekeeper, you've got a population of bees. You've got a colony of bees. Now that colony of bees, maybe in a season, maybe they supersede the queen once, and maybe they swarm twice. So you've got that colony of bees producing 3 viable queens, 3 queens that are going to become reproductive queens in the larger world. But that same colony is going to produce thousands of drones. So I like to talk about this concept of queens being deep and narrow, meaning that this mother colony that's going to both be producing queens and drones, that the queens that it produces are going to head a colony, and every worker in that colony, and we'll just talk about workers for now, half of the genetics of every one of those workers comes from that queen. So she has a very deep influence on the genetics of that hive, because half of every bee in there is from her genetics. But she's narrow, but it's a very narrow influence, because you've only got 3 colonies being headed by queens from that mother colony. But you have the opposite with drones. The drones are shallow and wide. You've got thousands of drones going out. They might be influencing the genetics in hundreds of hives, but maybe only 1/15 of the workers in that hive, in each of those hives, are influenced by the genetics from that mother colony. So I'd like to get that out of the way. So this is I took this from a presentation that I did at MIT a couple of years ago. And this was one of those stunts where they tell you that there's a fixed format where you have 20 PowerPoint slides and 15 seconds of slide to explain your thing. And this was actually a crafting form, so it was mostly people talking about quilting or baking pies. And I was talking about bee breeding, and I talked about how that, you know, if you're going to practice a craft like breeding bees, you have to understand your medium. And the medium for breeding bees is the genetics of the honeybee. So this is the genetics that we learned in high school. Like rabbits or people or bean plants. You have your girl bunny that has, and you have to start somewhere, so we're starting with red and blue. Your girl bunny that has red genes and the boy bunny has blue genes. The girl bunny produces an egg that has half of the genes that she has. The boy bunny produces a sperm that has half the genes that he has. And of course, your girl bunny and your boy bunny presumably love each other very, very much. So they share a special hug. We get our baby bunny. That's how we understand genetics to work. And I have to say, I don't remember when I first learned about the honeybee genetics. And I can't imagine that I didn't think that whoever was telling me this was putting me on, because nobody ever told me that things could work like this. So again, this was mostly designed for non-beekeepers. We all know what a queen looks like, and a drone looks like, and a worker looks like, and what the sting around a worker looks like. You know that very well. So workers make up about 80% of the colony in the active season. Of course, right now in this season, they probably make up, closer to 99% of the colony. We certainly see some drones in our colonies in the wintertime, but not a lot. The workers do all of the actual work except for laying eggs and mating with queens in the hive. Workers generally don't lay eggs, and we know that there's such thing as a laying worker, and we know from more current research that, that it happens a little bit more often than we think. But generally, the workers aren't laying eggs. Workers are nutritionally stunted queens. The difference between a queen and a worker is the amount of protein that she's getting and the kinds of protein that she's getting. And that a worker just doesn't have that stuff that makes their sexual organs and their sexual characteristics develop to the point where they can mate. Drones are produced from unfertilized eggs, which is weird, isn't it? It's like, going to the supermarket and the chicken eggs in the refrigerated section hatch into roosters. Like, that isn't how things work for most life forms that we know about. But drones are from unfertilized eggs, and I'll show you that in a second. When a drone mates, their penis explodes and they die. And all of the sperm in a given drone is identical. And not only is all that sperm identical to each other, but it's identical to the drone. Remember that drones are from unfertilized eggs. There's not a pair of chromosomes. There's one set of chromosomes in a drone. So what you see is what you get. There's no interplay between this dominant gene and this recessive gene, or these two dominant genes that are combining. The genes that describe the number of dark bands on the abdomen that the drone is carrying is displayed by the drone just by looking at it. Again, queens and workers are genetically identical. The same egg, the same larvae can produce a queen or a drone. The queens are fed a worker. What? I'm sorry, thank you, Ramona. A worker. The queens are fed a more protein-rich diet than workers. It was pointed out to me that they also get more sugar than the workers, but that sugar is there to get them to consume the more protein. That it really is these specific proteins that help the sexual organs develop. And the queen mates with between 8 and 30 drones in the first few weeks of her life, and she stores and nourishes that sperm to up to 5 years. That queen doesn't go out and mate again. 3 years later, 4 years later, 5 years later, if that queen is still laying workers, then that queen is using sperm that she's been feeding for 3, 4, 5 years and storing in her spermatheca, which is the organ that the queen bee has to maintain this population of sperm. So it's a little bit different when we look at what bees are doing, because the queen lays an unfertilized egg, just like everybody does, an unfertilized egg with half of the genes from that queen. But that unfertilized egg doesn't get fertilized, it develops into a drone. And that drone has just that half of the genetics from the queen. And then that drone produces a whole bunch of sperm, and that sperm is the same half of the genetics that is making up the drone. Now on the other side, it looks just like the bunnies that the queen produces an egg. Instead of the special hug, we have the fatal exploding penis. And we're talking about reproductive bees here, so we've got our new queen here, which is half of the old queen and half of the red queen and half of the blue queen. So what we see here, what you should be noticing, this is two queens mating. There's no male in this. The drone is like a flying sperm. The drone is genetically identical to a sperm. It's just a sperm that's grown up with wings. Whereas Kirsten Everson calls it, flying gametes or flying sperm. And this is the key that you have to understand if you're going to understand honeybee genetics, that these are two queens mating with one another that we're getting. Now, remember on the drone side, you've got a whole bunch of different ones, and we've got a mixed population, but we've got to focus this in somehow so that we can describe it and not get too confused. So in the next generation, we've got that queen, that red and blue queen that we just produced. She mates with a drone, which is a haploid clone of another queen. That haploid clone, that means the genes all come from that queen, not from anybody that queen is mated with. But it's not all of the genes, it's just half of them. The queen or the worker that comes out of that queen that's mated with that yellow drone is half from that yellow drone and half from the red and blue queen. But remember, it's not going to be equal slices. When we talk about half of a pie, depends on where you cut the pie, what is in the half. How many strawberries you get in that pie depends on how you cut it in half. It's good to be the one cutting the pie, knowing where the strawberries are. So it's not a fixed proportion of red and blue in the next generation. It's always a mixture. But note that the drone that's produced by that red and blue queen has nothing to do with the drone that queen mated with. There's no yellow in the drone. It's just the red and blue. What that means is that this generation of drone is a throwback to the previous generation. It's a subset of that successful mating that lived long enough to reproduce. It isn't a new combination like the queen or the worker. It's not additive, it's subtractive. Now, here we've got the yellow queen here, and that yellow queen produces these drones. But notice that each sperm from a given drone is the same half of the genes that the drone is. So we've got this queen producing these thousands of drones. Each drone has a different half of the genes, but each sperm that drone produces is identical. Now, when we have the yellow drone and the green drone, we've got all these drones from these different colonies mating with a queen. This is just a little expansion on the slide that we saw just a couple ago. That any of the drones that queen produces have nothing to do with the mating. But the workers and the queens are always a combination of the two, depending on which drone was the father, or which queen was the father, however you want to look at it. Now, these different colored drones, some of them could be from the same queen. They could share some genes, they might not. But those different colors could just represent different halves of genes from the same queen. But the eggs are different too. Each egg is a different half of the queen's genes. So we end up with this very interesting thing where we have the queen as a whole set of genes, and all these different subsets in halves of those genetics are producing these eggs. None of these two eggs are identical. But on the other side of things, we've got maybe 5 to 30 drones that queen is mating with, and each of those drones is providing millions of sperm for that queen. But you've really only got 5 to 30 variations of sperm. But the eggs are all different from one another, but they're all related, and some of them share the same genes because they're all different halves. The pie's cut in different places. And this is key to understanding things, that this really weird interplay of consistency and variation that's totally different from the classical genetics that we learn in school. And this is one of my favorite things about bees. I remember learning about this and just thinking it was just such a cool technique that the bees use. When the bees are queen right, when everything's going fine, the queen, she produces drones, she produces workers. The workers, depending on who she mated with. But what happens if we lose the queen? Well, we know what happens. We do a walk-away split, we lost the queen. And so what do? They make a new queen from a young larvae. But there's another term called hopelessly queenless. And hopelessly queenless is that there's no queen and there's no resources to raise a queen. If you went into your hive, I don't know, and we might be seeing some brood rearing happening right about now. But if you went into a hive right now and there weren't any brood in there and you killed the queen, either intentionally or unintentionally, or you put her in a cage, those bees are hopelessly queenless. They don't have those young larvae that they can make a new queen out of. So when that happens, you get the workers from this slide. See those workers? Well, they don't have a spermatheca. They don't mate. They can lay eggs eventually. With the pheromones from the queen absent from the hive, they've stopped being repressed from laying eggs. And they start to lay eggs, but they've never mated. So what do they make? They make drones. Now, drones don't do any work. All they do is take up resources and then fly out and try to mate with a queen. And this is what I think is just such a cool technique, is that when the hive is hopelessly queenless, the hive takes all of their resources and puts it into producing as many drones as they can until the whole thing peters out and dies. Until all the workers have died of old age and can't raise any more drones. And it's it's like diaspora. It's a last-ditch attempt to get as much of a chance of getting some of the genetics from that hive to end up in another hive before it just collapses on itself. So what? So all this adds up to a variety of genetic traits for the colony to draw upon, more than any two sets of genes could provide. Remember, we're talking about a social structure here. Can you imagine if you had to live with thousands of people that were exactly like you? I like to play the game of what would you talk about if you met yourself in a bar?
Bees.
Right, bees. But we tend to seek out people that aren't exactly like we are, and we tend to kind of think people were, I guess I should speak for myself, I tend to find people that are exactly like me a little bit annoying. I don't know what's wrong with them. So through this technique with having the multiple fathers and stuff, what it means is that we've got a variety of traits within this social structure that are all related and all are going in the same direction, but there's some variation in there to provide a little bit of relief from that monotony of hanging out with yourself or doing things exactly the way that you would do them. The other thing that happens here is that because of these multiple fathers and because of the drones and because of these haploid genetics, when a trait isn't immediately needed, like a trait that might be really advantageous in a cold winter or in a drought, that just because that doesn't happen one year, it doesn't get removed from the population. It tends to stick around in small amounts. I call this trait hoarding. It kind of sticks around in the background, and then you get another drought or another cold winter, and then those bees that survive, those are the ones that have been able to maintain those traits that are important. Now, I talked about Mendel because he figured out most of this genetics that we know about. And after he figured out the bean plants, which ends up being almost all the life forms that we know about how sexual reproduction happened, he tried to figure out bee breeding, and he failed. He does not seem to have understood haploid genetics. He didn't understand that the bees made it outside the colony. There were a lot of stuff he didn't seem to understand. But also he was a monk, and he was working under the purview of the church. And the idea that the queen would go out and have all these multiple matings probably had some political implications. And even if he could figure it out, he might not have felt free to express it. Certainly, a lot of smart scientists were thrown in jail by the church for being right. An interesting thing is that we have very good documentation that he did do this, that he tried to figure out bee breeding. What we don't have is any of his notes. You kind of wonder if they're in the basement of the Vatican somewhere. Okay, Okay, so now that we understand, do you have any questions about the genetics? Does that make sense? Yeah.
If you go back to the slide where there's a lot of colored circles and they're half and half, yeah. Could it be, I think it could be that there's a worker that's red, and what happens if she mates with some drone that's of red? You know what I mean?
Well, remember that the queen doesn't start laying eggs until after she's mated, so it's impossible for her to mate with her offspring. That can never happen. It is possible for her to mate with a sibling.
Okay, so say she mates with a brother who's red, right? And she's red. So what happens? Okay, one thing I talk about drones and the male bees being haploid, only having one, not a pair of chromosomes, but one chromosome. That isn't strictly the case. There is a gene, a loci on one of the chromosomes called the sex determination gene. Let's say that loci is where my fingernail is on these two fingers, and these are two chromosomes. There are something like 17 or 19 variations of this gene. This is a gene that lives at this spot always, but there's a whole bunch of different variations of it. 17, let's say. If they are different copies, if they are different copies of that gene, there's more than one variation of that gene, you get a female. If there's only one variation of that gene, you get a male. That's the sex determination. So that means that if the sex determination gene is the same variation in both chromosomes, you get a male. Now you've never seen that, because when that happens, that's called a diploid drone. It's not haploid, it's diploid. It has a pair of chromosomes, not one set of chromosomes. But it's a male. But the workers know pretty well that that's happening, and within about 6 hours of that transformation from egg to larvae, the bees chew them out. And that's one of the things that you see when you hear about inbreeding leading to a spotty brood pattern. That's what you're seeing. Now, if your spotty brood pattern is capped brood being, if you have open brood that's solid and capped brood that is spotty, that's not inbreeding. That's probably mites. It's probably bees chewing out mites or some other disease that's going on in the hive. Because they're going to chew that larvae out before you can even really see that larvae. Now in a laboratory you actually can raise a diploid drone. But the problem is that drone mates with the queen, and now you have a triploid offspring, and things go downhill very quickly from there. So, does that answer your question? Okay. Yeah. Okay. So raising queens, even if you're grafting on a large scale, even if you're raising a lot of queens, is not breeding. I know some operations in Florida that have been well known for generations for breeding bees because of the AHB fear in Florida. The bees that they're selling, they buy breeder queens from other areas, from other breeders, and they propagate them. You know, they have the breeder queens and they have the drone colonies and they mix them. They're no longer their own stock that they're raising. That's not breeding because you're not going from generation to generation. You're not refining, you're not selecting. You're buying stock and producing queens. Mixing and matching is popular but doesn't go far enough. And what I mean by that is, I'm sure, you know, not telling you, we've seen this over and over, and we all have these tendencies like, oh, well, I think I'm going to get, these Minnesota Hygienics, and I'm going to get one hive of Minnesota Hygienics, and then I'm going to get one hive of Russians, then I'm going to get one hive of these VSH bees from so-and-so, and I'm going to mix them all together and make my own bees. That's not really breeding until you start refining it, and I'll explain that in a minute. In a breeding program, no matter how small your breeding program, whether it's your 3 hives in your backyard or your 1,000 hives in Georgia, a real breeding program has to seek some kind of uniformity. And I talked about that before, that you've got to have some traits that you can point to that are the characteristics of your bees. Otherwise, there's no way to predict what you're going to get. There's no way to predict anything. And so, when you're going to start breeding bees, either start with what you've got, or figure out what it is that you think, and again, my criteria is about keeping bees without treatments. I don't care what your criteria is, just make sure you have some. If you think there's something better for what you want, other than VSH, and I'm going to talk about that in a minute. If you think there's something better than what you've got, go get them and start. But don't fall for the queen is always greener. We read the magazines, we read the forums, we talk to people, and it's like, we always think that there's these magic queens out there. I don't know anybody that's ever bought queens that was really an experienced beekeeper who thought that their purchased queens was the solution to their problem. The solution to the problem is breeding and refining. Because if you keep bringing stock in, you keep adding to the equation, you never get to that uniformity. You never get to that point where you have some characteristics that are working for you, because you're always bringing something new in, and you're never subtracting, you're never letting things weed out, you're never culling. Culling is just as important as anything else. You can produce excellent queens with almost any genetics if they're well-nourished and well-bred, uniform or not. But the problem with mishmash genetics is they offer nothing predictable for genetically for crossing. That means that if you're raising some queens, and you're raising some queens and you decide you want to, hey, let's collaborate, you can't say, well, you can't do that on the basis, well, I have these bees and they build up really quickly in the spring, and somebody else saying like, well, I have these bees and they're really frugal in the winter and don't eat any honey. That sounds like a great combination to try to bring together and to try to refine things with. But if you say, well, I've got these bees and they have this range of how well they build up, and this other guy says, I've got these bees and they have a range of how frugal they are, what do you get by mixing them? You don't know, because you haven't kind of refined things. So uniform stock has consistent traits that make it desirable for crossing, and hopefully you've selected that uniform stock, because whatever it is that they're uniform about is what's going to work for you and what's going to be helpful to you. So when you're breeding honeybees, you want to make an attempt to push things in the direction of the natural selection process. Inbreeding fixes traits. When more closely related genetics are bred together, you tend to fix those traits, those things that you want to be uniform, get fixed. Of course, you also start stacking up some less desirable things, and especially with the sex determination gene, you end up with spotty brood pattern. Outbreeding, breeding with totally unrelated stock results in vigor. That's why you should be breeding and you should be breeding. You should both be breeding together, so that you have something that you can cross and get that outbreeding. Because if everything's always being mismatched together, you can never achieve that outbreeding. Inbreeding is easy to detect in honeybees and easy to avoid. Just what I was talking about, that spotty brood pattern. And again, that's not spotty egg pattern and not spotty capped brood, but spotty open brood. Not uniform, really young open brood that gets spotty just before they start to get capped. You have to play a little bit of detective here. The nice thing is that when we see that spotty brood pattern,
What? Okay, we should be okay. It won't die quickly, and she'll get this sorted out. Even if you don't get to the point where you're really thinking about and analyzing exactly where they're spotty and why they're spotty, we fix spotty brood pattern almost the same way no matter what the cause is with brood queen. So you see a spotty brood pattern, it's consistently spotty. Unless it's the cat brood, you might be seeing some hygienic behavior that's removing mites. You might be seeing a burst of mites and the bees dealing with it. But generally when we see spotty brood pattern that we don't think is mites, it's a genetic problem. And we deal with it by replacing the queen. The other thing, consider introducing new stock via drones. If you have your bees and you want to, somebody else is breeding some bees and you like what they're doing, you don't really want to make half of the genetics in your hives all of a sudden from their hives. So you can take you can get a queen from them, and you can raise daughter queens from them. But remember, the drones don't have a father, so those daughter queens, the drones that those daughter queens produce, are going to be drones only from that other stock. So you can get some stock from somebody. Doesn't matter who they mate with. If all you do is are using them to produce drones to mate with your queens, then you're going to slowly and gently introduce some of these traits into the stock that you already have that you want. You don't want to turn, it's just like running a business. You don't want to change everything all at once. You slowly change things and you slowly let things evolve. But one of the most important things is that this breeding system, there's no way you're going to control it completely, and it works better when there's some dirt in the system. So don't be freaking out that like, oh, well, I can't control all the drones in my area, or I can't control this, or I can't control that. Yeah. Just go ahead and do it. Okay. I'm going to get to this because I think this is really important. So, this is a part of the talk that I think is really important because I think there are some myths out there that need to be dispelled. So I called this a localized breeding program, or is VSH for YOU. Everybody heard of VSH bees? VSH stands for Varroa sensitive hygiene. It used to be SMR. It's the same bees that used to be called SMR, which is suppressed mite resistance. VSH, you'll see them advertised everywhere. It's what everybody is saying is what you need so that you don't need to use chemicals with the mites and whatnot. So when queen breeders are breeding bees and selecting stock, they generally have a pretty long list of traits that they're looking at. How many frames of brood at dandelion bloom? How much honey did I produce? How big a cluster in the middle of winter? How much honey did they eat in the middle of winter? How many mites did I count at this date or at that date? There are all these parameters that we're looking at. But
The problem is that just like anything, when you start to apply a metric that isn't really what you're looking for, it's like standardized testing, then everything is going towards teaching to that test. And you're no longer actually testing for the thing that you want, which is ability or intelligence or academic, whatever it is. You're testing for that test. You're not testing for the things that really matter. So I'm going to tell you not worry about all these things, because there's 3 things that are important. I consider these meta traits or aggregate traits, the things that all of this stuff adds up to. There's survivability, there's productivity, and there's temperament. There really isn't anything else that we care about. Those are the 3 things that we care about. Now, we may have different, this order may change. If you're in a neighborhood with a bunch of kids, temperament's really important. If you're out in the desert with D. lesbi, temperament doesn't matter at all, it's just you and the cows. If you're making your living with honey production, then the productivity is really important. If you're trying to pollinate your blueberry garden, survivability is more important. If you're willing to buy bees once in a while to replace things, maybe survivability isn't as important as productivity and temperament. Maybe you like honey and you can't have your neighbor's stung, and you're willing to have bees that aren't quite as hardy. These are the 3 things that matter, and nothing else matters. Does that make sense? So before we talk about VSH, I'm going to talk about hygienic. And hygienic behavior and VSH are often confused. Hygienic behavior is, and again, this is something that people made up and is defined by the test that we use for it. Hygienic behavior that's the test that you might have seen in a book or a video once in a while, where they take like a coffee can and they put it over capped brood and they pour liquid nitrogen over that section of brood. And they basically freeze kill that circle of brood. They put the frame back in the hive. And they look to see how effectively the bees remove the dead brood from under the cappings. That's hygienic behavior. There's a lot of things that we think about as being hygienic kinds of behavior, but hygienic behavior with a capital HYG, that's how we measure it because that's how we've defined it. Hygienic behavior, it's not like it's either on or off, or you have it or you don't. It's kind of like height. Everybody has some height, but you might have a little bit of height, you might have a lot of height. There's a range, there's a continuum. So all bees have some hygienic behavior. There aren't any bees that won't ever remove any dead brood. Hygienic behavior, the reason it was researched and developed is because it can be effective against American foul brood and mites and other diseases, chalk brood. Because what that test shows us is how quickly the bees are willing to remove diseased organisms from the hive before they spread too much to the rest of the hive. The other thing about hygienic behavior is that it's not persistent over generations without specifically selecting for it. That means that if you start with hygienic bees and you want to keep that hygienic behavior, you have to actually do that test of freeze killing the brood or pin killing the brood or something to test for that hygienic behavior and use that as one of your selection criteria. Because if you just select for productivity, survivability, and temperament, which is really what we're all doing in the end anyways, that isn't sufficient to maintain that high level of hygienic behavior in the bees. Because remember, since it's present in all populations to some extent or another, what we're talking about is a very high expression of it. This is a hyper trait. A hyper expression of a trait that the bees have. Whoops. Now, when we look at the literature on hygienic behavior, when we look at American foul brood or mites, it's very much a brink effect. It means that if the bees remove 94% of those freeze-killed brood within 24 or 48 hours, depending on how you set up the test, they're going to be pretty resistant to American foul brood, and they're going to be pretty good at dealing with the mites. But if they only do 92%, it doesn't help. There's a line, you know. So it's really only this very tip of the hyperexpression of the trait that gives us the disease resistance that we're looking for. The nice thing about hygienic behavior is it's relatively easy to perform the assay to test for it. Again, you have to have some liquid nitrogen, and most of us don't have that sitting around. I don't, but it isn't terribly hard to you know, it's not like obtaining uranium or something. You can get some liquid. If you wanted to do this, you could do this. So now, Varroa-sensitive hygiene. The Varroa-sensitive hygiene, the research seems to indicate that it involves at least 4 different genes in the bees. It's not one trait, it's not like brown eyes or something. There's 4 different kind of genes that are interplaying that produces Varroa-sensitive hygiene behavior. And what the behavior is the ability of the bees to interrupt the reproductive cycle of the mites that are inside the cells. And usually that's done, the bees uncap a cell that has developing brood and a mite in there. And they either chew out the Varroa mite, or they do something else that interferes with the reproduction. So they might not kill the mite, but they keep the mite from reproducing, and that really is the key. And again, like hygienic behavior, all bees have some VSH behavior. It's a continuum, and all bees are somewhere on there. It's present in all populations. It's more specific to mites than hygienic behavior. Really, we're talking about mites here with VSH. Both mite removal and the prevention of mite reproduction are modes of action of VSH. And it's also similar to hygienic behavior with regards to persistence, that unless you are selecting specifically for that trait and doing the test to test for it, it's going to dissipate over the generations. And this is the key here. The problem is that testing for Varroa-sensitive hygiene is very time-consuming. And you're going to have Jeff Harris here soon, it sounded like, and one of the announcements was about having Jeff Harris at one of the meetings. Very smart guy. This is one of the things that he focuses on, and he'll tell you that they've tried to come up with some more efficient assays. But testing for Varroa-sensitive hygiene, instead of just pouring some liquid nitrogen on the hive and looking at it the next day, it involves uncapping brood that have mites in them and counting the reproductive mites. This is a very time-consuming process. Best guess is it takes at least an hour to do for each colony to do this once, which is fine if you're testing your colony in your backyard with a dissecting microscope. But if you've got 1,000 hives that you're trying to evaluate for breeding, all of a sudden things get out of hand really quickly. And this is the real problem. If you read the literature on Varroa-sensitive hygiene, in theory, the way it's supposed to work is you're supposed to test for it within your own population and select for it from within your own population. You're supposed to look at your bees and say, okay, my bees have this range of Varroa-sensitive hygiene, and I'm going to make sure that I breed from these ones that are right on the end here because I want this high expression of Varroa-sensitive hygiene. In practice, that isn't how it works. In practice, what happens is the USDA does these assays. They provide a couple of lines of VSH bees to some breeders. Those breeders produce queens from that. And the breeders that you see advertising in the magazines and on the forums and coming to try to sell you bees that are VSH, I don't know of more than 5 people in the world that have actually been doing the assay outside of the USDA. So all these, and I'm talking about, and I'm including in that the people that are very visibly selling stock that's VSH, that for years they've been selling stock saying that they have high expressing VSH queens that they're going to sell you, but they don't assay them for their VSH behavior. They are relying on this trait coming from the lines from the USDA and being this trait being incorporated that way, coming from the outside. So that's the wrong way to do it. It's not what the literature suggests is how it should be done. And it means that we're limiting, we're bottlenecking the gene pool a little bit in this VSH behavior rather than taking our populations and boosting the behavior a little bit. So there's virtually no source of VSH bees that the person selling them to you is actually testing them or even testing the parents to let you know, to assure you that those bees express the VSH behavior that is the basis upon which they are selling you the queens. Now, this isn't to say that this doesn't work. This isn't to say that for some of you, or even for all of you, that this isn't the right solution. The best way to use the system the way it's set up is that you buy VSH breeder queens from whoever you want to buy them from, somebody with a reputation for having good queens, and there's many out there. And you can graft from them, and you can make the offspring to your drones from your colonies that survived from last year that you bought from a different VSH breeder. right? Because you've got these survivors that survived over the winter. That's a good way to select your stock. They were VSH. You're crossing them with this year's VSH. And so you've got high VSH expressing queens, assuming that you started with high VSH expressing queens, that hopefully you did, but nobody's tested them to actually let you know that that's what happened. This absolutely seems to work. A lot of people have a lot of luck with it. If you want to use VSH trait for your bees to fight the mites, this is the way to do it. Raise your own queens, buy a new breeder or two every year, whether your club or yourself, however you want to do it, and breed this year's queens with last year's survivors. And you will have bees that can handle the mites better than if you didn't do that. No question, I'm not saying it doesn't work, but it's a little bit of a weird system. But here's the problem. We have a couple of clubs in our area that have done this, and I know in other areas of the country that this is kind of the obvious thing to do. You say, okay, I want to have a localized breeding program, and I want to reduce or eliminate my need for chemicals to fight the mites in my localized breeding program. So what I'm going to do is I'm going to buy some VSH breeders, and I'm going to breed them with my local stock, and that cross is going to give me my localized stock with VSH traits to fight the mites, and we've got our localized breeding program that is somewhat mite resistant or really mite resistant or whatever your expectations or the result is. That's the model, and that's what everybody does. But there's a trap here, and you have to be aware of this. So that's what you do. You buy a VSH breeder, you buy some VSH stock to raise your queens from. You enjoy the benefits of the VSH activity for a generation or three. You know, they do uncap the brood that have the mites in them. They do interfere with the reproduction, and your need for chemically treating the mites is reduced. There's no question about that. But the problem with that, remember that this trait dissipates over time, over the generations, unless you select for it specifically. And remember, there's a reason that nobody's actually doing the assay to do this, because it's really time consuming. So you don't do it and your local breeding program doesn't do it. So you get these few generations where you really get this nice bees that really don't require too much work as far as dealing with the mites. But of course you're skewing your selection. So now all of your bees, because they all have this VSH in them, all of your bees are doing a pretty good job at dealing with the mites. So when you're evaluating your bees, assuming you're not doing this VSH assay, you're looking at your survivability and your productivity and your temperament. You're no longer selecting for all of the other traits. We don't even need to know what they are. We know that they exist because not all the bees are dead. You're selecting against all of the other individual traits in your population that are contributing to their mite resistance, whatever level of mite resistance that may be. You're masking it. It's kind of like, you want to evaluate, 1,000 convenience store managers to see which the best ones are, but you give them all an extra $500,000 budget every year. Well, how do you know which ones can make the business work in their area? That's a different skill to actually be extracting money from the community and making it profitable and keeping that business in business than it is to take a whole bunch of free money and not lose your shirt. You no longer can find those shrewd individuals to train your next set of managers. You're diluting it, and you're not able to see where these other traits lie. You're not doing these VSH assays because it's too time consuming, and after a few generations, that hyperexpression moves back towards the mean, moves back towards average, and all of a sudden, no longer does that VSH give you the mite resistance that you were planning on. Well, you might as well have started with VSH at mean levels, because 3 generations later, that's where you're at. But the problem is that now you've got bees, now that that's dissipated back to the mean, because you've been selecting against all of the other traits that are providing mite resistance. Remember, some of those traits are probably metabolically expensive. They probably require some work on the part of the bees. It might impact your honey production. It might impact how they overwinter. It might impact all these other things that, while you're looking at the mite resistance with the VSH in the equation, you say, well, gee, I want more honey than that. But if without the VSH, you might say, well, they're not producing quite as much honey as these, but they're alive in the spring without mite treatments. Well, all of a sudden that reduced honey production actually means something. So now, you're a few generations down the road. You started this program by buying these $200 breeder queens to start with, and 3 generations down the road, you've got bees that are less mite resistant than when you started. And again, if you're going to bring in breeders every year to breed with last year's survivors, that's a fine way to do things. That's the right way to use this. This VSH is a tool, and that's the right way to use it. The right way to use it isn't to use it to start a localized breeding program that's going to be resistant, because it won't. So what should you do? Again, I talked about this before. Start with something. Whether it's your own stock or something you think is better, get it and start it. Select and breed your bees and rinse and repeat. You gotta keep doing this. Don't worry too much about diversity and bottlenecks. Bottlenecks are how evolution happens. Bottlenecks are culling out the stuff that isn't what you want. These are important things. And again, if it's too bottlenecked, you'll see that spotty brood pattern, and you'll know you're inbred and you have to do something about it. And some level of uniformity is the goal. So, I know everybody wants the same thing. They say, oh, well, our club should have a breeding program. Our club should be raising queens. But I'd rather see you raising queens, and you raising queens, and you raising queens, and you raising queens. 10 different lines, then 10 of you getting together and breeding bees together. Because if you got 10 different lines, you've got 10 different things going on, not one thing going on. Breeding is a process. There's no goal, only just improvement. You know, you're never gonna be done. Because even if you breed the perfect bee, you can't maintain those characteristics exactly the same way from year to year. There's always evolution going on. There's always some kind of pressure that's changing things. So once you start breeding, you're a breeder, and you're a breeder for the duration. There is nothing that you can buy that's good enough. I don't know a single knowledgeable beekeeper who ever bought queens and thought that that was the solution to their problem. Even if they don't raise queens themselves, they always want something better. The only way to get something better is to do it yourself. You can't fix a problem by turning the natural model on its head. Remember I talked about the deep queens, the deep shallow queens and the wide, deep and narrow queens and the wide and shallow drones. What you don't want to do is to say, well, I love this hive, this is my best hive, and then go and requeen all your 100 hives with daughter queens from that hive. Remember that model where the hive, you know, a good colony produces a few queens and thousands of drones. You have to maintain that kind of balance, because that's the system that works. That's the system that has let bees to be here for 50 million years. And everything boils down to survivability, productivity, and temperament. And I don't care how many mite counts you do, how many assays you do, how much you weigh your hives, how much you inspect. Nobody's come up with a metric that can predict these things, except for measuring them directly, because those are the things that we care about. And that's it. I think we've got a minute or two. Are there a couple of questions, or, yeah?
For the hobby beekeepers, some people are telling me that you should requeen every fall, all your hives, your few hives. What do you think of that? If you've got a strong hive, and it's been great, the queen can last for a year.
Well, I can tell by your question what you think of it. And my, you always want to hedge your bets. So maybe you've got a queen and you really like her. So before she dies and supersedes herself, or before you replace her with a purchased queen, raise some queens from her and hedge your bets. And then you've got options, and you're building on what you've got. Because if you've got a strong hive that you like, do you want to replace that? No, you want to build on it. You want it to get better over time, and it won't get better by replacing the queen with a mated queen. It will get better by raising your own queens from those queens. But don't make the mistake of saying this one hive is, I'm going to requeen my whole operation with just daughters of that queen. Now, with 3 hives, you can do that. Also, you should look at some of the stuff that Mike Palmer and Kirk Webster do up in Vermont with overwintering nukes. It's a little bit different, a little bit further south, and there's some things that are easier and some things that are more difficult. But there's overwintering nukes so that you have nukes in the spring to replace your dead outs with. Right, and keep everything going and doing your breeding before things die out. That's really the way to do it. So look up on YouTube, look up Mike Palmer, and you'll see it. Kirk Webster writes about it on his website on kirkwebster.com. Is there anything else? Yes?
What do you think is killing the bees?
Beekeepers.
Yeah, well, beekeepers have a lot to do with it. Remember that 99% of the bees are run by 1% of the beekeepers. 3/4 of the bees right now in the country are in California doing the almond pollination. There's a lot of chemicals in the environment. Not all of them are systemic pesticides. Some of them are fungicides. Also remember that insect populations tend to be boom and bust. You know, we see it with gypsy, we see it with all kinds of insects. Bees we happen to be focused on because we care about them. And we put a lot of pressure on the bees. We want to have, you know, millions of them we require millions of them to be alive and pumped up in the spring at this time of year to do the almonds, when that isn't what they do. So I think people is probably the most concise and most accurate answer. But bees die on their own too. But in nature, that boom and bust kind of happens on its own, and we're not observing it and not freaking out over it, and we're not contributing to it. I don't know if that is a helpful answer or not, but you know, yeah, and lack of good forage is important. You know, we've changed a lot about how we manage the environment. You know, we used to, I know where I grew up, they used to burn the fields every few years, so there were these meadows full of wildflowers. Well, I go back there now, and a few trees have grown up, and now it's to the point where nobody's willing to cut down a tree. So once the trees show up, nobody wants to cut down the tree unless they're going to put a development in. And neither the forest nor the development is as good forage for the bees as the meadow that could otherwise be there. Anything else? Yes?
I have a sort of a process question. When you were talking about splits earlier, and you would say leave the small, or say a nuke, and leave it for the foragers to come back to.
Mm-hmm.
Is it sufficient for the hive, the main hive that you've moved? Will enough foragers come back and find that one wherever it is?
Yeah, the bees actually have an interesting feedback mechanism where they know how much brood is in the hive, and they know how much protein and nutrition is coming in, and they will reallocate. You know, assuming there's a good population of bees in there, they will reallocate nurse bees to become foragers before their time, so that they can repopulate that. And hopefully you're doing this with a hive that's strong enough that if there isn't a whole lot of food coming in for a couple of days, they're not about to starve. You know. Most of the foragers will go to the original colony, and the colony that you've moved, if it's a strong colony with a good population, will very quickly reestablish a good forage population from what it has as resources. Does that answer your question?
Yes.
Okay. Okay, I think that's probably all we have time for, but we're here. Oh, sure. Yep.
The genetic thing was kind of mind-boggling for me. Like, if you would have a couple of hives, what is the chance of inbreeding with just a couple of hives?
Well, remember that drones drift very prolifically, and they fly, you know, miles and miles and miles. If you have I guarantee that if you have two hives and one of them is flying out queens for mating, that most of the drones that she's mating with are not from your hives. Now that may be a little bit different in a very large concentrated apiary. We've been in apiaries with 800 hives and seen queens falling out of the air surrounded by drones. They couldn't get out of the yard without getting mated with. But in a smaller apiary situation, I don't think it's as much of a problem. But again, you'll see it in a brood pattern. It isn't like barn cats where you kind of over generation see them start to look a little bit weird. The bees, it's very clear what's going on.